Antibacterial sensitivity profile of Lactobacillus species and characterization of aflatoxin producing fungi isolated from yam and plantain flour. This article is written by 4 authors. First author Rosabro Praise Timmy Lade. Second author Rolatan Janet Ayubakoa. Third author Wahab Abidine King Kunri. Last author Hamdalu Wafan Malayo Rukayat. Contribution and originality of the study is that, this study documents the inherent probiotic bacteria isolated from yam and plantain flour. These microorganisms enhance the improvement of the gastrointestinal tract. Therefore, storing these food products in a humid-free environment will prevent moisture accumulation and aflatoxin contamination. This in turn reduces the risk of foodborne intoxications. This study investigated the beneficial effects of fermenting microorganisms as well as the presence of aflatoxins in milled flour samples. Thermotolerance, pH tolerance, phenol tolerance, temperature sensitivity, in vitro antimicrobial potentials of lactic acid bacteria, LAB, isolates and aflatoxin detection in the milled flour sample were analyzed. Lab isolates were identified and characterized as Lactobacillus species using standard procedures. Probiotic properties and antimicrobial activity of lab isolates were investigated using standard methods. In addition, quantitative and qualitative detection of aflatoxins in the milled flour samples were also studied. The results showed that all the lab isolates were Lactobacillus species which survived in high temperature range of 50 C 60 C, acidic pH 2, 3, 4 and 5, phenol tolerance, 0.1%, 0.2%, 0.3% and 0.4%. In vitro antimicrobial properties of the lab species, SO1 and SO4, shows that they were able to inhibit the growth of Escherichia coli ATCC 43816, and Klebsiella pneumoniae 25922 with the corresponding zones of inhibition, 25 mm and 20 mm. Due to the effect of storage time, 6 months, yam and plantain flour samples moisture content yielded 9.50% and 9.20% with a corresponding total number of aflatoxins, 859.3 and 899.0. In conclusion, lab species isolated from yam and plantain flour samples have probiotic potentials. Moreover, the longer the storage time, the higher the number of aflatoxins. Thus, they should be consumed within the shortest time possible to avoid caking and moisture accumulation, which tends to decrease the number of beneficial microbes present in them. Thanks for watching this video of the International Journal of Biotechnology Volume 9 Issue Number 1.